Hey there folks, welcome back to our final two lectures, Synapses and Neurotransmitters. I decided to make these into one lecture just for the ease and brevity of doing this. So you can watch this all at once, or you could break it up into two lectures if you'd like, but I don't think that it's going to be too burdensome to just watch the entire lecture here. So let's move on. Now, remember, we have gotten through structure of neurons, electricity that is happening within our neurons, graded potentials, resting membrane potentials, and now action potentials. And so here we are at the axon terminal of a neuron. And we are going to look at today, how does that one neuron, this presynaptic neuron, communicate with the postsynaptic neuron and stimulate an EPSP or an IPSP. Now, that synapse is a junction between two neurons, or it can be a junction between a neuron and a sensory cell, or it can be a junction between a neuron and an effector cell. Okay, so we can have these different types of synapses, and it just depends on where we are in our bodies. So two neurons communicating together, a neuron and a sensory cell. So if that sensory cell is communicating with a neuron to fire, or if that neuron is firing and communicating with a muscle fiber to tell it to have some kind of output or effect on our body. Now, there's a couple of different types of synapses, just so that you're familiar with this. There's an axodendritic. So an axodendritic basically means that the axon is connecting with a dendrite of the postsynaptic neuron. We can have an axosomatic, where the axon of the presynaptic neuron is synapsing or connecting with our soma, or the cell body of our postsynaptic neuron. Or we can have axoaxonic, where we actually see a neuron that is connecting at the axon of another neuron. Now, if we look at the communication between those neurons, there's two ways these neurons actually communicate. And as you can see here in this diagram, we have our presynaptic neuron connected to our postsynaptic neuron via its dendrite, so an axodendritic connection. And it's relying on our good old friend gap junctions here. And so we can call this an electrical connection. There is direct contact, and the ions are passing directly from cell to cell. So that sodium that's coming in is passing directly into here to trigger another action potential, or an EPSP in this case, and then eventually an action potential if we reach threshold. Now, you can imagine there's almost no delay in this type of communication, okay? But if we look at our chemical connection, we see that there is a neurotransmitter that's released from one cell that is released into that synapse, or the synaptic cleft, a space between the two, and it binds to receptors on the postsynaptic neuron. And so there can be a slight delay of, a half to 4 milliseconds. A couple of examples then, with an electrical synapse, we find these in parts of the body that are dependent on defense reflexes, whereas our chemical synapses would occur at the neuromuscular junction between a neuron and a muscle fiber. This is a nice image of a synaptic knob that is connecting here to the soma, so axosomatic connections with all of these synaptic knobs coming in here. So you can see that there's a lot of information coming from other places in the body that are going to come into this one soma. Now, what do chemical synapses look like? And we're going to spend most of our time talking about chemical synapses. So they have a presynaptic cell. they are using neurotransmitters, and these chemical synapses allow for a little bit more variability in whether or not they fire or not. So we have neurotransmitters that are packaged in vesicles within those presynaptic cells. There's a synaptic cleft, or a small little gap between our synaptic knob and our dendrite, or our postsynaptic cell. And then on that postsynaptic cell, we have receptors for different neurotransmitters. And if those neurotransmitters can bind to those receptors, well, then we're going to have some kind of opening or closing of those receptors. And then here's our postsynaptic cell there with those receptors. Now, those neurotransmitters, when they're dumped into the synaptic cleft, they need to be removed or reused or recycled or broken down. Otherwise, they would just continue to fire on those receptors or bind to those receptors. Now, we're going to look a little more into detail on this, but you can see this image is showing our signal coming down our presynaptic neuron. That's going to stimulate this vesicle. It's there, it's primed, it's ready to go, and it's going to fuse. It's going to release the neurotransmitters. They're going to bind to our receptors. Our receptors are going to maybe open, and that's going to allow ions to pass through, and that's going to have either an excitatory or an inhibitory effect on our postsynaptic neuron here. And let's just put this up. Now, you will need to know this for your test. You need to know the pathway that is causing the release of these neurotransmitters. So first, our action potential is being transmitted down to our axon terminal. That triggers voltage-gated calcium channels to open. So if our voltage-gated channel opens, calcium is going to rush in to our presynaptic cell. That calcium then signals our vesicles to move to our membrane. Any vesicles that are docked and ready are then released by exocytosis. And as those are released, new vesicles are making their way to redock and get prepped and ready so that they can release their neurotransmitters if there was another signal coming down. So that neurotransmitter is then going to diffuse across the synaptic cleft and bind to a receptor on the postsynaptic cell. That receptor should open and allow ions to move into this postsynaptic cell here, and that's going to cause an EPSP or an IPSP on this postsynaptic cell. So here are those EPSPs. If you remember, they're going to bring us a little bit closer to threshold, and so we're becoming a little less negative or a little more positive, and that makes it more likely for the neuron to fire and have an action potential start at that trigger zone. In order to make that happen, we usually see sodium rushing in and making the cell membrane slightly more positive. If it's an IPSP, remember we're moving farther away from our threshold, and so it's going to require a stronger signal or make it less likely to fire. And this is usually chloride ions that are moving into our cells. So if neurons are getting more messages at once, you can see in this diagram that we're getting stimulus, another stimulus, another stimulus, another stimulus, another stimulus. We can see that it gets closer to threshold. It starts to die off a little bit, but we get another stimulus and it brings us up a little bit more closer. It dies off. We get another one, drops down, another one, it drops down, and eventually we hit threshold. So we can call this summation, adding up our postsynaptic potentials and responding to our effect. Now, we can also have one neuron that's receiving EPSPs and IPSPs at the same time. So maybe that is getting closer and then farther away and then closer and then closer and then farther away. So we get this sort of summation of these EPSPs and IPSPs. So you can see in this image here, we've got a couple of stimuli that are bringing us really close to threshold, but oh man, this neuron fired and it just brought us back down. And then it kind of starts to return, and then up we get another signal from these neurons. That brings us closer, and then this one fires and it takes us farther away, and then we get a really big signal to send in and change the membrane potential of this cell. And so this is happening at that axon helix. When we talk about our trigger zone, this is where we need to have our membrane reach -55. Now there's a couple different types of summation. So the first one is temporal summation. So you can think of this as time, temporal for time. So here you can see that we've got 3 neurons, and they are interfacing here with this postsynaptic neuron. But in this case, we're having one neuron that's sending some action potentials pretty rapidly down its axon. They're going to hit this soma, and those EPSPs are going to have an additive effect on that trigger zone and bring this cell to -55 to threshold, and that cell is then going to then fire down its axon. So remember, temporal summation is time. You can think of this as more signals in a shorter period of time that are going to have this additive effect to trigger our postsynaptic neuron. Spatial summation, then, on the other hand, is having stimulation from multiple different synapses in different spaces. So spatial or space, and so you can see all 3 of our neurons in this example are sending in signals, but not very quickly, but all of their effects together bring this neuron to a closer to threshold, which causes this neuron to fire its signal. So these neurons may be cooperating, or when we talked about converging signals, this is a really great example here where we're seeing multiple inputs coming in to stimulate this neuron into firing. Now, once those neurotransmitters are released, we need to remove them or reuptake them. You can see here that a neurotransmitter only binds for about a millisecond. If you remember, these molecules, they have some energy, and so what happens is they can tend to vibrate loose from that receptor. Other times, we have astrocytes that will help clean up our synapses here as well. If we didn't turn off this signal, that postsynaptic neuron may continue to fire. So if we leave those neurotransmitters in that synaptic cleft, they're going to continue to bind to receptors and open those channels and cause EPSPs or potentially IPSPs, and we get no firing. And so that could be a bad situation for our bodies. Again, that neurotransmitter dissociates, but if it's not removed, another one's going to bind, and then another one, and then another one, and another one. So we have to stop that signal, okay? So we can, one, just stop releasing neurotransmitters from that presynaptic neuron. So if there's no signals coming down that presynaptic neuron, well, there's no more neurotransmitter that's being released. But what about the neurotransmitter that's left over? So one, it may just diffuse into the nearby extracellular fluid, and you can see diffusion away from our synapse over here. It may be reuptaken by proteins that are in that plasma membrane that are going to reabsorb those and bring them back into the cell and recycle them and repackage them into new vesicles. Or we may have enzymes that degrade them as well. And in this case, we're going to talk about acetylcholinesterase. So before we move on, pause the video, answer these questions, practice this material a little bit, and I'll see you in a few minutes. All right, welcome back. Let's move on. So now what we want to do is talk about our categories of neurotransmitters. What kind are they? Now this is sort of the beginning of lecture 16. If you're feeling like you want to take a break, come back and we'll go over neurotransmitters the next time. Otherwise, we're just going to keep moving along here with neurotransmitters. So what are the effects that neurotransmitters have? Well, we have said already that some can be excitatory, meaning that those neurotransmitters open channels that allow for positive sodium or positive ions to move into the cell and bring that postsynaptic cell closer to the threshold. Some can be inhibitory. So either chloride ions are moving in, or maybe potassium ions are moving out, and we're making that cell a little bit more negative, and therefore farther away from threshold and less likely to fire. Some of them just kind of depends on the postsynaptic cell receptors, we'll see. So the same the same neurotransmitter can bind to different types of receptors. And those different types of receptors then open different channels for different ions. Some of those neurotransmitters open ligand-gated channels, and we'll see that some act through a second messenger system. So a neurotransmitter will bind to a receptor, and that'll set off a cascade event that will trigger these second messengers to have other effects on the cells, sometimes changing protein synthesis, or maybe opening other ion channels. So these neurotransmitters do not have the same effect everywhere. So as I said before, there's multiple receptor types that can exist for different neurotransmitters. So for example, serotonin has 14 different receptor types, and these all have different effects on our postsynaptic neurons. And so you might have an excitatory effect in one part of the body, where you have an inhibitory effect in a different part of the body, but you're still using the same neurotransmitters. And so again, it's dependent on which neurons are firing and which pathways they're firing along. So you might not have serotonin being released all over your body. It's more localized to a certain region, and that's going to have different effects in our bodies. Now the receptor governs that neurotransmitter effect on that target cell. Now, if we look at our neurotransmitters, our first one we're going to talk about is acetylcholine. So this is part of the cholinergic system. It's kind of in a class all by itself. So you don't need to know the chemical formula for acetylcholine. Now, cholinergic neurons, or synapses, use acetylcholine as their neurotransmitter. And so this is going to control the contraction of muscles. So there's two main types of receptors. We have nicotinic receptors and muscarinic receptors. Nicotinic receptors bind to nicotine, and muscarinic receptors bind to muscarine. And so that's kind of where they get their name, was that they were sort of identified with those chemicals binding to them. So if we look at nicotinic acetylcholine receptors, they are ligand-gated channels. Okay, so over here we can see we've got acetylcholine. We actually have to have two acetylcholine neurotransmitters bind, and that's going to open these ligand-gated channels. And in doing so, we see sodium moves down its concentration gradient, and potassium moves out and down its concentration gradient. If you look at this, we're going to produce an EPSP, so that means more sodium has to move in than potassium out, so that we get a change in our voltage closer to threshold, or getting more positive. And so we see these receptors in our brain, our autonomic nervous system, skeletal muscles, so all over. With our nicotinic Receptors, then again, this is just another sort of a zoomed in and cleaned up version of that image. So our acetylcholine binds to these channels, sodium diffuses in, potassium diffuses out, but the overall effect is making the cell more positive and bringing it to threshold. A little closer look at this system then. If we have our presynaptic neuron and action potential is arriving and it's triggering those voltage-gated calcium channels to open, then that is stimulating the exocytosis of those docked vesicles that are waiting there. Our acetylcholine diffuses across and binds to those ligand-gated channels. We see that sodium is rushing in and potassium is rushing out, and that is going to eventually cause our cell to depolarize and send an action potential along that postsynaptic axon. Now, muscarinic receptors, or muscarinic acetylcholine receptors, is an example of our second messenger system, and it utilizes what's called a G protein. So we can see here that our acetylcholine is binding to our receptor. It's activating this G protein, and that G protein actually opens a potassium channel, and the potassium then diffuses out, and it creates an IPSP and has an inhibitory effect and hyperpolarizes our postsynaptic cell. So we see this in the brain, smooth in cardiac muscle, and in different types of glands. So again, an inhibitory effect with muscarinic acetylcholine receptors. Now, basically that neurotransmitter is binding. Just to kind of show you a little more detail here, the neurotransmitter is binding, which is then activating our G protein. You don't need to memorize alpha, beta, gamma protein or the GDP, but what I want you to know is that our G protein becomes activated. It is then opening another ion channel, in this case, our potassium channels, okay? So here, then potassium flows out and we have an inhibitory effect. So in our heart, we can slow our heart rate down with acetylcholine. Now, acetylcholine esterase is an enzyme that breaks down acetylcholine in our synaptic cleft, and therefore it inactivates it. So you can see in this diagram, we've got this big chain of events that is producing acetylcholine. Acetylcholine is released through our synaptic vesicles into our synapse, where it's going to bind, but acetylcholine esterase breaks that into acetate and choline and basically deactivates it. Its reuptake, we have the choline is taken back into the cell and reused to make more acetylcholine. Now, if we inhibit acetylcholine esterase, all right, and with nerve gas, well, now we leave acetylcholine in our synaptic clefts. And we just mentioned back here that this causes our heart rate to slow down. So if we're talking about muscarinic receptors in our heart and there's nerve gas present, the acetylcholine is going to continue to bind with those muscarinic receptors, progressively slowing down and slowing down and slowing down and slowing down that heart rate until eventually it stops. Now, go ahead and pause your video, practice with the material we just went over, and I'll see you in a minute. Okay, so how'd it go? Hopefully what happens if acetylcholine esterase is inhibited, and we talked about nerve gas. So again, be trying to apply these principles to the greater view of our understanding. So if we're inhibiting excitatory neurotransmitters or inhibitory neurotransmitters, what effect might that have down the road? All right, moving on with our exploration of neurotransmitters, our next group is called the monoamines, and these are some neurotransmitters you've probably heard a lot about, so serotonin, dopamine, norepinephrine, and histamine. So again, our monoamines then are our epinephrine, norepinephrine, dopamine, histamine, serotonin. These work through G protein coupled receptors, so they're going to have that second messenger system. With those second messenger systems, it really depends on the neuron, but there's all kinds of different results that can happen from these neurotransmitters in the second messenger system. We get signal amplification. We can open and close other ion channels. We may activate enzymes or activate genes to increase protein synthesis. So there's a lot of different end results with these with these neurotransmitters. Now, if we just take a little closer look at this group of monoamines, you can see that the monoamines are produced and stored in those synaptic vesicles. They're produced from amino acids here. And so we get our end product, or our neurotransmitter, being stored in these vesicles. They're released into our synaptic cleft, and they're taken back up by transporters or proteins in that presynaptic cell. And in this case, we can see that monoamine oxidase, MAO, degrades those monoamine neurotransmitters into their individual components to be recycled and reused. MAO can be blocked by MAO inhibitors, so these are often like antidepressants that help to increase that neurotransmitter's effect. So this is one way to keep that neurotransmitter in here longer and allow for an excitatory response on that postsynaptic cell. So serotonin regulates mood, behavior, appetite, and the circulation in our brains. You can see that it kind of is used for mood, memory, sleep, or cognition. And if we have too little, this is when we can become depressed or have aggression, become a little bit irritable. And as we'll see when we talk about drug use, if we flood our systems with serotonin, then we generally will have withdrawal of serotonin or a decrease in the production. It takes a while to make these, and so this is kind of where this irritability and aggression and depression can come from after heavy drug use. Dopamine, again, controls movement, emotional response, pleasure and pain. In Parkinson's disease, we have the degeneration of those dopamine-releasing neurons, and that ends up causing these involuntary contractions. Norepinephrine, we see the use of this, norepinephrine and epinephrine, in our fight or flight responses in our sympathetic nervous system. So this is going to stimulate us, get us ready to get amped up and fight if something's coming at us, or run away as fast as we can. And then histamines tend to be involved with our wakefulness, learning, memory, and they respond to injuries and allergic reactions. So if any of you guys have had allergies, you may have taken some of these in the past to kind of tone down those histamines. Now our next group are our amino acids. So we can see glutamate, glycine, and GABA are sort of the three that we'll be talking about. The first one we'll look at is glutamate, and this is a major excitatory neurotransmitter in our brain. So we see this located in our cerebral cortex and brainstem, and again, this is involved in learning and memory, which is really important for anatomy and physiology. Next is glycine. This is the most common inhibitory neurotransmitter found in the spinal cord. We see that in the brain, in the spinal cord, and our retina. And GABA is our most common inhibitory neurotransmitter in the brain, and we'll see some examples here of glutamate and GABA when we talk about mouse party. So we said GABA is an inhibitory response. So how does that work? Well, if you look at this image on the right, we can see that here's our presynaptic terminal. We have calcium rushing in. We have GABA that's released that binds, and GABA is going to open chloride channels. So chloride is going to rush in and make this postsynaptic cell a little more negative and less likely to fire. So it's going to be inhibited. And finally, we have neuropeptides. So neuropeptides are chains of 2 to 40 amino acids. They generally are released in combination with other neurotransmitters, and they inhibit pain being sent up to our brains. So let's look at Mouse Party here really quick. This is a website, and this is in your syllabus. We're going to look at meth, cocaine, and ecstasy, and I want you to look for some similarities and differences between the three, and then the rest of the drugs I want you to work on at home. Basically what you're looking for is, again, that application and the analysis of what's going on with these drugs, what's going on in our synapses, and what's going on with our neurotransmitters. So what is that interaction between the drug and the neurotransmitters, or the neurotransmitter receptors, and what kind of effect does it have on the body? And you can expect to see some questions on the test about that. So let's go ahead and come over here to our Mouse Party. Now, you will need Adobe Flash Player for this. So if you need to download that, or if you need to use a computer that has it. So basically what you're going to do is, we got this cool little aquarium full of drugged up mice. One's drunk, one's sitting over there, he's on cocaine. You can see he's got a little white on his nose. We got this guy with LSD, he's just kind of zoning out on his hand there. So let's start with meth here, and we're going to put him in our chair. So you're going to click on him and grab him and drag him to the chair, and then we're going to analyze what's going on in his brain here.
Dopamine transporters are responsible for removing dopamine from the synaptic cleft. Because meth mimics dopamine, it is taken into the cell by the dopamine transporters.
Okay, great. So first thing, we're talking about dopamine neurotransmitters. We can see we've got dopamine receptors down here in our postsynaptic neuron. And we have our dopamine transporters here. So they're going to be bringing dopamine back in, but what we saw was that meth mimics dopamine, so it's brought in by those transporters.
Once inside the cell, meth enters the dopamine vesicles, forcing the dopamine molecules out.
All right. So now, what do you think's going to happen if we're forcing this dopamine out and those meth molecules are staying in these vesicles? What do you think is going to happen next?
The excess dopamine in the cell causes the transporters to start working in reverse, actively pumping dopamine out of the cell and into the synapse.
Whoa, right? So now this cell is basically releasing all of that dopamine out here where it's not supposed to be building up in content, right? Or in concentration. We just talked about how our bodies are trying to eliminate and clear those synapses. So dopamine is now flooded into our synapse.
The excess dopamine becomes trapped in the synaptic cleft. As a result, it binds again and again to the receptors, overstimulating the cell.
Right? Remember, that dopamine normally would be taken back into the cell by these dopamine transporters. And at this point, we have reversed the effect of those transporters, and so dopamine is being pushed out, and so we're stimulating and stimulating and stimulating and stimulating again and again and again and again and again. And so you can imagine the excitatory signals coming into these postsynaptic neurons.
Meth is highly addictive because it works directly on the brain's reward pathway, making the user feel intense pleasure and exhilaration.
All right, great. So we're talking about our reward pathway here. All right, so let's get rid of that mouse. And let's look at our Coke mouse. All right, Coke mouse, have a seat. He's our movie star mouse. Just kidding.
Dopamine transporters are responsible for removing dopamine molecules from the synaptic cleft after they have done their job.
Okay, so here we are. Very, very similar beginning to what we saw with meth. We're talking about dopamine. We're talking about those dopamine transporters.
Cocaine blocks these transporters, leaving dopamine trapped in the synaptic cleft. As a result, dopamine binds again and again to the receptors, overstimulating the cell.
All right, so a little bit different than meth. So instead of actually going into the cells and going into the vesicles and forcing the dopamine out and flooding a bunch more dopamine into our synaptic cleft, this drug, cocaine, just inhibits those transporters, so dopamine just stays in that cleft.
Like other drugs, cocaine concentrates in the reward pathway. However, it is also active in the part of the brain controlling voluntary movements. This is why cocaine abusers are fidgety and unable to be still.
So when you think about that, we've got these voluntary movements. So if we're getting EPSPs on neurons that are triggering voluntary movements, right, it almost becomes sort of involuntary because we're exciting those neurons and we're kind of fidgeting and moving around and rubbing, and right, and so you get this sort of involuntary control of our bodies because of that excitation, because the dopamine transporters are blocked and dopamine is remaining in that synaptic cleft. All right, last one that we're going to look at here, which is our ecstasy mouse. Look at him dancing. Oh man, they should have him with like lights on his fingers and masks on his face or something like that at a rave. That would have been pretty cool.
Serotonin transporters are responsible for removing serotonin molecules from the synaptic cleft after they have done their job.
All right, cool. So now we're talking about the same situation as we saw with meth and cocaine, but now we're talking about a different neurotransmitter. Here we're talking about serotonin and serotonin transporters.
Ecstasy mimics serotonin and is taken up by serotonin transporters. In fact, ecstasy is more readily taken up than serotonin itself.
All right, so it's a serotonin mimic, and it gets picked even before serotonin does.
This interaction with ecstasy alters the transporter. The transporter becomes temporarily confused and starts to do its job in reverse. The transporter starts transporting serotonin out of the cell.
All right, so again, we kind of saw this with meth, a little bit different, but again, that there's this release and flood of serotonin coming out of that presynaptic cell into our synaptic cleft. So you can imagine what's about to happen.
The excess serotonin becomes trapped in the synaptic cleft. As a result, it binds again and again to the receptors, overstimulating the cell.
All right, so we're getting lots of EPSPs on those
Ecstasy
postsynaptic
affects serotonin
cells.
pathways responsible for mood, sleep, perception, and appetite. Ecstasy also indirectly interacts with the reward pathway. The excess serotonin stimulates a milder release of dopamine along the reward pathway, giving ecstasy slightly addictive properties.
Right, so when we think about that, and hopefully at this point you've maybe talked about the brain a little bit. I don't know if we've gotten to that part in labs yet, but you're going to see mood and sleep and perception all affected and heightened here, right? We also see how serotonin stimulates releases of dopamine. So now we're not only increasing serotonin, but we're having this slight release of dopamine in this reward pathway. And so now we take this, we get dopamine into our reward pathway, and we're like, oh man, I take this pill, I have this reward, and we reinforce that connection and make this drug a little bit more addictive. All right, now the rest of these mice are yours to explore and check out. You're gonna have a hard time waking this guy up. He's kind of zonked out there. So you can go ahead and practice this last slide, review and reinforce all of these different neurotransmitters, receptors, and bring in how drugs are going to be affecting them.